So our mission is to get from there to here without spilling.
And are there any clues in that chemistry for thinking about how active this region is? I mean, considering that previous visit in 2009, was it? Any changes or sort of clues that we're looking for in the active elements of this chimney?
Like
Something has changed. I'm just looking at the paper that was, there's only two papers published, and I'm just looking at the paper, and a sentence here is on-board study of the recovered chimney, which is from right about here, revealed that it was composed almost entirely of colloidal sulfur. So just white, yellow sulfur, but that's not, doesn't appear to be what we're seeing here. So something has evolved.
like I remember. Oh yeah. Oh
Yeah, I mean yellow.
yeah.
Right.
Just pretty much native sulfur. Yeah.
Yeah. We're looking at the pictures
Yeah.
of the older chimney and looking at this. So that's why I'm surprised.
Mackenzie, can you tilt down a little?
Not what we were expecting. But
it may or may not go anymore.
So that's what they recovered.
Keep going,
sort of looks on the outside like the same thing, but when they got it on the ship, it's yellow sulfur, native sulfur.
Wow. Yeah.
So the fluids may have evolved. I mean, that's what it feels like, doesn't it?
yeah, I think, you know, I Kamawegu Seamount, so formerly known as Loihi in Hawaii, had an eruption that happened in like 1996, 1990, sometime in the 1990s. And what they found in the vent fluids is that it was temporarily enriched in like iron and manganese.
Okay. You can,
and so I wouldn't be surprised if
and,
this might have been
HD extend.
maybe not the case here, but
then,
where you have some magmatic input
and the camera
or an injection that's delivering fresh iron and manganese to these hydrothermal fluids and enriching them in those metals.
So I'll let you and Ed work together to. So you have to tilt up a bit.
That's pretty good.
Yeah.
I'm going to come in a little tighter,
Swing
just about the
swing
wrist.
it left a little bit.
And Brianna, do you have any special, any squid facts? Because we do have some serious squid fans joining us.
More, Ed.
Coming in, about there.
I think one squid fact that I think is cool is the cockeyed squid, how it kind of swims on its side because it has one eye that is bigger and meant to actually like look up,
full zoom there,
you know, for predators, and then the other eye is a little bit smaller.
That's what we want. Happy with this one here, Brina?
Yeah, looking good.
Wait, so a Roger on the second sample?
Well, and will you be able to tell which
one is which? Yeah, not me. They all look like shrimp and rocks, but we can put it in a different area of the box. Or you could put it in the other box, maybe. It could be, if you're... Hey, get off my jaws. There's nothing to eat there. Science huddle.
Would it be possible to bag more shrimp since they're all pretty abundant here? I know the contact station there in Saipan would definitely appreciate having something like this to share with the public.
Yeah, Roger.
Right.
Are we going to do another grab here?
Yes. Yes, another grab here.
I'm teasing shrimp meanwhile. Attempt another grab. I should have quit while I was ahead.
That little...
Nope.
That wiggle you did last time worked really well. Oh, there you go. There you go.
Oh, man, look at it. Look at this. Yeah, look.
That's gorgeous.
Okay.
Oh, you can see that.
Wow.
Yeah, you're right. It looks like Galena, doesn't it? pyrites.
Twitch
Galena, some sulfide. Wow.
I wonder if there's...
Here, let me look up.
Be interesting to see if sphalerite is in there or something like that.
I'm going to bring it right towards you, Ed, so don't freak out.
Jacob, you know that there are sulfides on the arc at East Diamante. There's a large field of chimneys, black smoker chimneys, and mostly extinct now. So. And it was the only place on the arc that sulfide chimneys, black smokers, were found. So this is a new.
Yeah. Unprecedented. That's really interesting.
Because at northwest of Fuku, it's those sulfur chimneys. Oh, look at the limpets on this as well. That's great. I was hoping to see some of those. These little guys. Right. Those are limpets.
The science manager's going to hate us. 87 subsamples.
Well, it's a little cone it's a snail that is pyramidal shape or got a curly bit on the top and just sucked down on the rock. And they're also bacterial feeders, and a few of them. Well, one species does have chemosynthetic bacteria in its gills, but that's not on at this site. This is probably Lepidodrillus, just recently described, Lepidodrillus marianus, and it is one of the few species that's found in the back arc and on the arc as well. So it can cross that interesting chemical divide.
I was wondering if we could lower this sample a bit so I could get a shot with the still camera, unless you think it's better to kind of be more cautious and just put it into.
Or can we zoom out? Would that help?
I can put it in the still camera.
Yeah.
What is that?
We'll get a really nice shot.
Yeah.
Yeah.
Yeah, it's close there. No guts, no glory.
Verena, we also had some viewers wondering about those shrimp. For people that are just tuning in, what is it that they're all doing there, and why so many?
Okay, so yeah, I did do a little expose on the shrimp earlier, but let me just recap. So there's what those shrimp are telling us is that this rock pile, this chimney that looks like it's not venting, is releasing some fluid, because that fluid is sustaining bacterial growth, the white stuff that's coating this chimney, to the point where it's growing fast enough that it can support this population of shrimp. These shrimp are, they're grazers, like they mow the lawn of the little shrimp of the microbial filaments that are growing up. So they're crawling all over each other, trying to get to a little spot where they can cut some grass, so to speak.
Gorgeous, thank you.
That's what they collected.
We'll leave it on there, Mackenzie. Do you want to roll a little bit there in the camera there? Did you get that shot?
Yeah, who you talking to?
Science. Oh, you'll make me nervous if you're doing anything right now, because I'm literally balanced on a marble with picking my nose with the manipulator. So I'm doing this. It's taking quite the bandwidth. So when I see you reach down here, I think you're doing something, and I go, ah! It just catches out of the corner of my eye.
Oh.
Until we get it in the box, I don't relax. Did you get that shot?
Yep. Got it.
One more, or are you happy?
Let's do one more, and then I think
make sure you get the Nautilus logo in there, and
yeah, exactly.
Dr. Robert Ballard.
All right. I think we're good.
Okay, and we're gonna put this in the front, right, with the other one? Or
yeah, try to put it into the left side of the bio box. Yeah.
Okay, Mackenzie, if you pan the HD camera to the right and tilt down again. I think Ed was correct. We never did rack out. Just tilt it down till I can see the black bit of the top. Yeah, perfect. Thanks. That mid camera really, like, there's parallax there. Right now it looks like I'm really close to the box, but if you look in that camera, I'm like miles above it. Parallax error. Like, where are you really? I don't know. That's why I usually reach out and touch something with the manipulator. Or conversely, try and go in somewhere without touching anything. It's like that game, yeah, where you get electrocuted when you... Former boss hooked up a shocker to my chair, and every time I touch it...
When I started in film production, our cameras had viewfinders that you couldn't look through the lens. So they were offset. You had to kind of mentally compensate for the parallax.
Yeah, it was painful, eh?
Oh, man.
Okay, Mackenzie, if you forward box close, going to be your top right button there.
Gotcha.
Oops, stop.
Oh.
Let me give you a little help here. Okay, try again.
A little snag on something.
Oh.
Yeah, wait, it's our 20-year-old design. Okay, try a little close.
Oh.
There you go. Now you should be able to close. If you open the box all the way, it's only supported on the N2 rollers. And then when I smash on it, when I jump on it with a manipulator, it can actually bend that one roller. It's only, so that whole box is held there by a 1/4-inch bolt going sideways.
Oh, I see.
Okay, you can close it all the way. You'll see in that camera when the lid closes.
There we go.
I'm just gonna, I'm gonna try and fix this annoying little twitch here, so give me 1 second.
Is that twitch when you lock it? Is that what I'm seeing?
It's coming on and off the freeze. So.
Huh.
Always one. Well, we saw the clams. Right. Yep. Oh.
That is a servo valve offset, so
yeah.
I'm putting a...
I'll be off comms for a second. Pilot video.
Yeah, go ahead.
Science is asking if we're able to zoom on the fisheye.
Stand by.
Oh, somebody's headset. fisheye cameras visible on our satellite feed 3, channel 3, on the nautiluslive.org website.
Roger.
Mm-hmm. Yep. Have you looked at the chimney samples in the forward lounge?
Okay.
The chimney samples in the forward lounge. Okay.
Where were we?
There's
The upper camera. That is.
in a case in front.
Let's say 8 to 11, right?
I have the zoom control for that.
Yeah.
And where we typically run that for the super secret, yet to be released photogrammetry is here on mid-zoom. So that's an engineering level of some thing that you probably know about that's over my head, but that's...
Is that full zoom?
No, that's a little bit off of mid-zoom, so it's some lens thingy.
Oh, it's a preset, right? That's the .7 or whatever?
Yeah, it's from my world, it's a command of -7 to the servo, but in Jonathan's world, it's
11 millimeter?
Some number that doesn't make any sense to me. Sorry, I'm not a shutterbug.
We love the technical talk, Jan.
And it's supposed to, I'm supposed to put in a thing that puts it back there after 3 minutes. So we're given the leeway to zoom in or out at our discretion while we're sampling, but during general operations, he's requested to have it at this -6 or -7 or whatever.
Did you want more of a zoom with that science, or was that what you wanted?
Yeah, so
we can.
You can't go in. I don't know.
We can zoom in, but then I have to mess around and focus it.
Oh,
so
yeah.
The focus is kind of set for this
Okay,
level.
well, that's fine. Let's not worry about it.
Yeah, so zoomed in is just to show you what it looks like that.
That's pretty good.
But then I have to
spot focus.
Yeah, I have to pull up the controller and focus it.
No, look, it's not worth it then. That's fine.
And I do that occasionally anyways if I, but I'm using it for a pilot camera to see the manipulators and look at the structures and gross kind of things. The same applies to all 3 of those cameras. So the camera we call mid is that one. He also wants that at
-7.
7 or something in, yeah. And then the lower camera he wants zoomed in so it doesn't see any of the jewelry, but obviously now we have jewelry on the porch and the porch out. So we're aware of this as some trade-offs we've made operationally. I think the request was to slurp some shrimp.
Yes, please.
Okay. If you could put the jar camera, Ed, where the winch is currently.
So we have a sample in which jar now?
I don't know. We're gonna work on that.
It should be. We filled jar 1. It should be on jar 2 because they just rotated it 1 jar.
Is that right?
Yeah.
Roger.
Okay.
And then Mackenzie, if you could go to your camera page and turn off the rail camera and turn on the jar cam or bubble cam or whatever it's called.
Starboard and bucket.
Bucket cam, yeah, that's it. Beautiful. Gotcha. Do y'all want to, happy to just flush through this one or flush or what's, I don't know.
I think you can go right into that one because we just indexed it and after we stopped suction, so it should be ready to go.
Roger that. Let's zoom in on some victims over there.
Volunteers.
Volunteers, that's a better word. Go ahead, Ed. We can push right in there.
Yep, coming in.
So we have a target-rich environment here.
Holding for a second while I sort something out over here. Coming in.
Roger.
Hold it about there as you go in.
No, go. Give me full zoom there.
Oh, that's right. You got more.
Is that, I'm going to extend the camera again.
Sure. That's going to need a bump down.
Roger.
Okay, I have a question.
You can full zoom.
A friable sample. This is not fried fish that we're collecting. What is a friable sample?
breaks apart.
Like a snow cone.
Just you try to grab it, and it just crumbles. So yes.
Friable, yeah, easily broken apart.
Got it.
It's one of those highly technical geological terms, you know.
I see. And so these biological samples, do you want to talk a little bit about how they're processed here on the ship, and then where they go from there? Maybe Brianna too, you can jump in if you want to talk about that.
Yeah, so after we collect, especially the biological samples, a lot of them will get processed in the wet lab that we have on board, and we will probably preserve them in ethanol, and then they'll get shipped back to the Museum of Comparative Zoology, and then their scientists across the world are able to use them for their own research, whether it's for morphology, looking at their characteristics, or for molecular work to do any sort of genetic comparisons. And it's also very useful because we know exactly where these animals came from and have the exact depth and temperature and all this video footage to show their environment that they're in. So this is really great for research.
They learn pretty fast, don't they?
I'm impressed with your suction, though. They zoom right in there.
Yeah, there was also a question in the chat about what effect the difference in pressure has on the biologic samples. So these shrimp, when they are brought on the ship, are they still live? Or are they affected by that journey from the bottom to the top?
Usually they're in a pressurized container of some sort so they can decompress properly on the way up. So they are fortunately alive on their way up, and then they meet their demise somewhere at the top. But yeah, that's usually there's like technology that we've learned along the way to help keep them at a similar pressure to where they're currently found at.
That's not the case with the suction sampler, though.
Can I jump in for a second?
No, it's suction sampling.
That's the amount of shrimp you have in the jar?
Yeah,
Wow.
that's looking fine. Yeah, no, that's great.
Okay, Mackenzie,
we found that
shrimp are escape artists, so we need to rotate that jar.
Most animals from about 1,500 meters to the surface will stay alive. The point here is that they have no no bladders, no air pockets that can expand on the way up. What does happen is that sometimes dissolved gases in the blood or fluids of an animal's tissues will come out as solution from really deep water, and that can be the problem. But we've been able to keep a lot of these animals alive on the ship, looking at their behavior, looking at looking at what they're doing alive. Yeah.
You were saying?
That's the flush jar, yeah? Okay.
Yeah.
And I know, Sean, you mentioned too the value in having this kind of sample for thinking about communities that have shallow water shrimp populations too in the Mariana Islands. You know, I'm curious what the sort of comparative benefit might be too for thinking about these marine life that live at the depths, and then how that sort of is related to those more shallow species.
Do a little housekeeping here.
Yeah, back in Saipan, we don't have a lot of saltwater shallow shrimp species per se. We do have some brackish water ones that kind of live inland in these non-existent rivers that we have. So you know, when we see shrimp in our waters, you know, this might be the best example of you know, some of the shrimp fauna in the Marianas, and bringing it back on shore into the station, and helping to, you know, share, and just have folks kind of pick it up and look at it. You know, I think it's very beneficial. And just understanding how large or small these shrimp are, you know, is also
put our, I forgot I had it
interesting,
over
and
here.
it should be a
You
good
can put the winch back up
conversation
there, Ed.
piece, and
Winch
looking
quad or winch
forward
alt,
to
your
sharing
choice.
with the community.
In this image here, you can see more clearly how these shrimp are working the surface, and underneath their heads are specialized little claws that they're very small, but they're picking off the filaments. They look very busy. The smaller ones are juveniles that are probably about a year old. We have some idea they probably live about 3 years. We've been tracking the populations at Northwest Rhoda over our 6 visits there, and we've seen mass recruitment of little tiny babies that have come down out of the water, and they're only maybe not even they're about half a centimeter long, and then we see them grow up pretty quickly. The females appear to have an annual cycle, probably reproducing, releasing their babies somewhere in winter time, then the larvae, so they hatch out of the eggs. The little larvae hatch out of the eggs that she releases, and the larvae swim up in the water, and we don't know anything about how long they are up in the water column. Probably not all that long, because they don't want to get washed too far away. But really, it's just not known. And then they recruit back down. They feed in the water column, so they feed on photosynthetic productivity, and then they come down to the bottom and start feeding on the chemosynthetic. And you can actually see that in the carbon and nitrogen isotopes in the bodies of the shrimp when you analyze it. And as the shrimp get bigger and bigger, there is a shift from photosynthetic carbon and nitrogen isotopes to chemosynthetic carbon, because the carbon 13 to 12 to 13 ratios differ. It's kind of complicated, but it's kind of a wonderful tool to know how much food is coming from the sea surface source from photosynthesis versus how much food is actually coming from the hot vents. And then you analyze the muscles of the animal, the tissues of the animal, and you get a pretty good idea of how dominated it is by just chemosynthetic production when you're now looking at these adults.
I have a question for Verena. So I've seen, you know, when we're talking to communities, we have pictures of vents that have maybe are a little bit more diverse biologically. You see like shrimp and mussels and crabs and other things. Like when you see one that's just full of shrimp, like what strikes you? Is this like a emerging or a going away system where the biodiversity kind of changed to?
Yes, I think we're seeing actually a very reduced amount of fluid flow here, so we are not seeing a very extended habitat, and it's not a habitat that's suitable for a lot of animals. I'm a little surprised not to see the crab that is present at every other Mariana Arc vent site, and it's not here. I'm just wondering, and it's one of the reasons why this site is interesting, it's what can be attracted here and what can live here, and what is the characteristics of the fluid that allow certain of these animals to be here. Now, at the beginning of the dive, we saw clams. Nowhere else on this arc do you find clams. So what is it about this site that allowed those clams to survive? The tube worm that lives here doesn't live anywhere else on the volcanic arc. So there's something peculiar about this particular site that is attracting different species, and the barnacle as well. On the previous shift, they saw a lot of barnacles. But that barnacle isn't anywhere else on this arc. So there is something intriguing about Toto caldera, and one of the reasons to look at it today is just to get a better idea of what the community looks like. And there's another cruise coming back here in December from Jamstec, and working with them, it would be really fun to put together the information from both cruises to characterize this. But your question is a really good one. Each volcano on the Mariana Arc tends to not have much very high diversity.
Brina, just interrupt for a
Yes.
second. Should we move upslope?
Yes,
And then
we
you
should.
can continue
Sorry.
your story.
Yes, I'm getting a little carried away.
That's all
So
right.
it's a good time to move.
But we can multitask.
Do let shall. So
Mr. Dan. He doesn't have his headset on.
yep, they're talking nav over there.
Okay.
Yeah, I was just discussing with Lynette. There was a request from somewhere to put bathymetry on the ROV machine here.
Yep.
And historically, that machine is working pretty hard already with all the track lines. And if we try and put bathymetry on it and along track lines, it will become bogged down, and eventually it will require intervention from either Lynette or DE, which will interrupt operations.
Doesn't sound good.
No. And besides that, it's very distracting for me. I'm used to, I'm an old dog. I'm used to a black nav screen with track lines on it, like even the long track lines. So every time I glance at it, it takes me an extra 2 or 3 seconds for my reptile brain to process what's happening with the ROVs and the ship. And I, crash into things.
Okay. Well,
in the future, what we can do is Lynette can work her magic with HiPAC.
yeah,
And
that sounds like a good solution.
Yeah, for the next dive, we can fill your boots.
Okay, so we're ready to move from this site.
Yeah, where are we going?
I think we want to move upslope a little bit.
Roger, uphill. I like uphill.
Look around. We still haven't found the source of the really white, cloudy water that we encounter sometimes. So I think our main goal is to try to find where all that's coming from. So there should be some sulfur chimneys emitting white smoke somewhere in this area. And I think we're going to try to just traverse kind of up and down the slope, offsetting to each side a little bit, and see if we can run across them.
Roger. Yes, please.
I also kind of wanted to keep our eyes open as we're making our way up the ridge for any potential rock samples. Might be good to add to the starboard bio box, but
yeah, we're late. We're late, by the way, so
oh yeah,
we need rocks. We need
in the vent area. They're all coated in sulfur, though. But if you want a pristine rock, we'll have to wait till we depart and get out of the venting area.
Yeah.
Did they pick a rock up at the beginning of the dive?
We picked up one rock,
as
far as I know.
So
one little bitty rock.
Unfortunately, it was just a bunch of talus, but we did get one. So
okay.
Hey, this is Nav. So in northwesterly direction, upslope. Does that sound good to everyone?
Yeah, give me a minute to get my bearings. We haven't moved at all since I sat down, so I'm gonna stretch it out a little and look at that next lump in front of us.
Yeah.
We'll come up a bit, see how high it is.
Yep. Look at them all. Now that's impressive.
yeah, definitely some shimmering.
Yeah.
Wow.
Dan, is that an air bubble on the Zeus lens? Do you see it?
So there's actually a fair number of deposits
could
here.
Could be oil, could be on the inside. I'm going to come up and get a close look at this guy here.
Yeah.
Did you
I heard
see,
it as I was getting up.
You said you saw venting? I don't see any shimmering water.
It's all right. We can just keep moving. It's that.
Oh, roger. Shimmering over here.
We want the cloud.
Roger.
We're looking for.
Hunt down that cloud.
We're looking for white smoke and this sulfur chimneys.
Ooh, there's a nice little fisher.
Oh,
yeah.
Wow.
Okay.
Oh, some tube worms. Dead tube worms.
Oh yeah.
Let's try 20 meters north, Anna. Please 20 meters north.
Could get a nice.
20 meters north, Roger.
Yeah, see if we can hit that chimney.
Oh,
with
there's
that
like a
lens.
little. It's down there.
I'm teasing Anna, see if you can hit the chimney with that lantern.
Looks like that drop.
That's unkind.
From here.
Huh.
This looks healthier.
Just gonna look around here. We're looking. I can't tell if they're alive or not. Verena will speak up. Ooh, smoky stuff.
Aha. Is that something we stirred up or?
Hard to say. I don't think so, but
Yeah.
it's possible.
I think it may.
looks like
I,
ROV.
yeah, it
boiler.
does. It does.
Oh, okay.
While we were setting down there, we were probably making a dust cloud behind us.
Uh-huh.
That's probably it floating up the hill. Mackenzie, you can come up 5. And tilt up. Get Herc in the camera there. Sorry, I come back out of my dust cloud, so we'll come around the other way here. I did see a little diffuse venting maybe coming out from underneath. Are there any known flange pools in this area? Or is
No.
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 Dan loves it.
No, we never seen anything like that. Then the big chimneys in the back arc, it was not forming flanges.
All right.
There, the chimneys are Endeavour-like
Yeah.
in terms of size.
I'm kind of at the end of my leash here, so waiting for the ship to catch up.
Okay, we got.
Do you want to? Are you tempted to sample these, Verena?
I am.
We'll get it.
Okay. I think we might as well.
Okay, let's just go in and see what we've got that might be alive.
Roger. Okay, I'm gonna have to.
It would be the ones that are hanging over an edge.
Come down 5 meters. Yeah, down 5. It's a bit of a rough landing, but I'm pulling on the Atlanta a bit. Okay, Ed, find us a live tuber.
Yeah, I'm looking. I think I might have.
You can
No,
push in
that's
a bit
shrimp
there.
on the end of one. I'm looking.
See anything there, Brina, that looks interesting?
No,
Don't have any.
I did see live ones when we first came down on our first watch. If you remember, there were a few that
Yeah.
I was pointing out the red
Yep.
tips, but
Uh,
not here.
push in just a bit, Ed, and we'll
Yeah.
strafe the cliff here. Yeah, that's good. Oh, we're looking for red stuff?
Yeah, but you have to go slowly because
Right.
it's too fast.
I'll slow
I
down.
just need to scan them
I'll get a little closer here.
because some of the red is actually other animals
Or
here.
shrimp, yeah. Yeah,
I
they all
don't
look
see.
empty, don't they?
Yep, they sure do. I'd say this is no-go. It would be too much time and struggle with not much
Yeah.
return.
I'll just, while we're waiting for Atalanta, I'll swing around the rock to our left here, where it's overhanging, and I can't go too far.
So these worms, to grow to this size, we're probably fairly old because they're not getting a, looks like they're growing pretty slowly.
When you say old, is that
Well,
decades or eons?
how long does it take one of these to grow to this size? If there's not really vigorous fluid flow, it can take them a couple of decades.
Wow.
But these guys they're not seeing any more fluids, so they're totally dependent on the fluids coming out, and they need hydrogen sulfide. So there must be sulfide in these vents.
Yep.
Fluids, or else you wouldn't get these, and the clams the same thing. Which, why didn't I put 2 and 2 together before? That would make sense.
There's a little bit more here to my right. I'm being a little gross here because I'm pulling on Atalanta.
Dan, should I do anything else to adjust Atalanta?
Nope. It's, you might see your thrusters bite a little as I pull on you here.
No, don't see it.
I think if we're not going to grab any tube worms in this area, some of the rocks here don't look too bad, so...
What, like this over here or something like that? Well, it depends where he can stabilize, but...
Yeah.
I can stabilize anywhere. Well, I can try.
Okay.
I might have enough leash. We can look. It looks like the structure continues above us, so I can come up a bit if you want.
If you see something, just go for it.
Or we can try for one of these rocks.
Like that rock.
I do. Yeah. I like that rock.
Okay. Telestrator.
Yeah, it's wonderful. Where's it been all our lives, Bill?
Yeah.
None of this stupid pointer, you know, you're trying to smash on the screen in front of the pilot.
Yeah.
And it's their excuse to get us out of the front row, too.
There's 7 you guys in the front row.
We still have the pointer up here.
We do. I used to take it away from Rennie and beat him with it.
Right.
Almost like it's fasciculated.
Yeah.
It does, yeah.
We had to keep the telestrator private for a year until people got tired of drawing cats.
Pushing it a bit
Yeah,
on
a lot of the rocks around here
on the rock for us.
are pretty vesicular, so. Ooh.
Beautiful.
Get you a little more
empty tubes.
contrast here. Where's my focus going? Are you drifting? Yeah.
Okay.
I'm moving around a little with the minute here. You have to come wide a little. I'm not sure where it is. Thought I had my camera centered.
Me?
So is there any correlation between that red that we see with the shrimps and the red of the tube worms?
For sample or
No.
just zoom?
No,
Both.
Yep.
the reddish color on the shrimp has to do with
Push in just a little more.
their egg production or their gonad, and for some and I'm not sure what the pigment is. The tube worms, what we're looking for, red plumes, is hemoglobin, blood. For the chemosynthetic animals, the best pigment is hemoglobin, which you don't usually find in most deep sea animals. They like hemoglobin because it can grab oxygen really well in this oxygen depleted environment. It's the best blood pigment. It also can grab sulfide. That's especially a special kind of hemoglobin, not like ours. It has 2 sites on it where the oxygen goes on one side and the sulfide clicks in the other side so they can't react. And then that's carried to the symbionts inside the worm.
I always
And
thought
then
it just grabbed
those
the
guys,
hydrogen sulfide and not
then the
oxygen.
little magic energy happens.
Wow.
And we can discuss that a little bit later, what exactly how chemosynthesis works.
Looks like part of it's definitely a little altered, but most of it is pretty good. So
Looks good.
All right, got a good rock.
I'll move it down into the cinema camera if you want. Ace digital stills camera.
Would that
all right.
Hopefully.
would that be better in the?
That's a beautiful shot with the shrimp and the two berms all going around the rock.
I wish I had more zoom there for you. I think I'm full.
Or you could put it in the starboard box, just closer to the front.
Yeah.
I read it. Mackenzie, you can change the rail cam to the whatever cam.
Gotcha.
I can never remember the names of those things. Yeah, that one. And you can slide that box open if you want. You happy with the shot there? You want rotate or?
Yeah.
Do we get lasers on it yet?
Not down there.
No, we did not get lasers down there. So what we need is more lasers.
But the jaws are, I don't know, maybe I'll get lasers on it down there. I doubt it.
All right. I think one more still shot and we're good to go. Perfect.
Got it?
Right. Yep.
Yeah, Dan, could that go in the armpit box? I mean, what? Sorry, you call it the starboard?
Yeah, there's the lasers on it just for scale.
Thanks.
I call it the starboard and the forward.
Okay.
It might be called some other wonky name, but for me it's starboard, forward. And we were going to put it in the starboard box, right?
Please.
And then we're going to
just a little concern that we don't want to break the chimney samples.
Yeah. This will be the second rock in the starboard box.
Yep, that's right.
Oh, look, the other one is still where I left it.
This is like scratching your back.
Yeah.
That's a good analogy.
Oops.
Okay.
Good. You got a rock.
Rock number 2. We need more rocks. We're late. We went in thinking Bill and I would be greedy, and so far we've been really conservative. Little tiny rocks.
Are you guys okay with moving on?
Yeah.
Okay, so I think from here,
Major samples.
it would be good to, I don't think we want to go shallower from here. We probably want to go jog to the left, say, about 10, 15 meters.
Roger.
So that means southwest.
Okay,
and then,
you can go in.
And then kind of back down slope again, down to 2980 or something.
Roger.
And try to cover that ground.
I was just going to pop up and see if we were at the top here.
Okay, take a look around.
But, yeah.
So we're still looking for the source of the really cloudy water. Presumably that looks like little sulfur chimneys, but you know, it's going to be.
I was hoping it was at the top right here.
Yeah.
But I don't know. Smoky. Flying by Braille. That looks like the top. There is a structure approximately 10 meters to my starboard. And it could be where the smoke is coming. I have a leash to run over there real quick.
Okay.
I'm looking at sonar targets on the smaller nav screen here. I'm flying like a crazy person.
There's a lot of...
It looks like it's coming from this way.
Yeah.
Flying by braille again. Oh yeah. This is, there's structure, more structure here, so I'm picking up more structure.
Yeah, there's a lot of steep slopes and open fissures.
Yeah, where are we at? Yeah. I am.
What is all that white particulate that's in the...
It's sulfur.
All of that sulfur? Like
Mostly.
This is, seems to be the top here, Bill. There's
Okay.
nothing on the sonar.
Ed, could you put the high pack screen on this lower monitor for me?
Absolutely. You're at the work table?
Yeah.
Gotcha.
I don't know where that's coming from. Just look around here and see if we can.
I'm going to start with 2.
There it is. Thank you.
There we go.
Can you tilt down on the HD, Mackenzie?
HD, yeah.
Yeah, it's really slow down, but you have to hold it.
All right, so Dan, it'd be great to kind of lateral to the southwest about 20, 30 meters.
Southwest 20, 30 meters, roger. Like east, south. I still get those confused. Waypoint 5-ish?
Yeah, and maybe
Okay.
a little past it,
Roger.
but going through it.
Yeah. Stand by. Stand by. We'll get Atalanta. We'll get Hercules over there where we move the boat.
That really cloudy water really comes and goes.
Yeah, I thought it was, I really thought it was coming from the structure. And you can kind of see the fissure there
Yeah,
in,
you can.
in the sonar above
Yep.
Anna. So we're looking at that. Oh,
Look at those rocks.
look at those rocks.
Yeah, I can see rocks.
Yeah. And they're not covered in
I'm
sulfur
glad there's
either.
somebody in the room that loves them.
Adam, if you're listening, we're parroting you.
That's a kind of been a
Kind
marker.
of, I'm backing down the fissure here to the southwest.
Okay.
Looking for smoke. Yeah,
Lots of shrimp.
lots of shrimp.
Yeah.
Making a bit of a dust storm as I go backwards here.
Well,
Is there a beeping sound that's
yes,
happening?
I was
Is it-
wondering.
That's why we have the hydrophone turned off right now.
Yeah,
Sounds like a garbage truck back there.
yeah.
What do you see on it?
Can we have a little more delta, perhaps?
Yeah, we're all right.
Talus, all of a sudden.
Yeah, I'm going away from the structure there. Can look to your left a little bit. See if we're tangled up yet.
So which sub-track line do you
Yeah, you can.
find more
That's right. You can
reliable?
come up just 5 meters, nice and easy.
I assume the red one is USBL and the white one is DVL. Is that right, Dan?
On
on
which
the
screen?
high-pack screen?
I don't know. I don't look at that screen. It's
Yeah,
green and lines to me.
the red is DVL, and the, I don't know, pinkish, grayish, whitish is USBL.
Oh, okay. I had them backwards.
So we're just guessing between the two, are we? Or is one more reliable than the other?
The USBL is more accurate in an absolute sense, probably, because
Yeah,
the
it
DVL
sort of
drifts.
depends. If there's like a bunch of hot plumes and things like that, we can get more refraction errors. But I think the DVL has been drifting a fair bit down here too. So
Do you do you make the DVL coincide with the USBL periodically, or?
yeah. So periodically we'll reset the DVL position to the USBL position.
Yeah.
We like to do that when we've been sitting still for a while.
Right.
So we have a nice kind of reliable cloud. Yeah.
What is it to the southwest you're after?
Just to cover some new ground
Roger.
and our little search pattern.
Roger. It's downhill that way, but
it's okay. We've got some smoke coming, so.
Yeah. Have we sat still and tried to see which way the smoke is blowing?
Yeah. It seemed, last time I did, it seemed to be coming from the north, the way we were going. Well, that was like last watch.
Okay, yeah.
I haven't done it. I haven't paid attention this time. That's a good idea. Mackenzie, if you, what's my tether turns action here? What happens if you bring your head to the right, look northeast?
I think we could head downslope now, if that works for you.
Yeah.
Did you want a ship move that way, southwest?
Yep.
Okay, well, maybe 30 meters southwest.
Since that's downhill, I'm gonna hang out here.
Yeah.
And right now I'm pulling Mackenzie tail to tail. Our delta's good, but our altitude is danger, danger.
Yep.
As you pointed out, thank you for that. So we can hang out here and kind of look. I'm not sure how far Bill wants to go.
Down to about 2980 depth, so that's more or less due south of the ship, probably.
Roger. So since we're going downhill, we'll keep Atalanta on top of Hercules or in the deeper water, which is going to be out here somewhere.
Gotcha.
And,
okay.
When's the last time you
maybe
operated this thing?
Last time I operated what?
This thing.
100 years ago?
Really?
Yeah.
I wouldn't go that far west. I'd go a little more southwest if you're talking about moving the ship, because we're gonna, Hercules is gonna now go southeast downslope.
Yeah.
Okay. So maybe 30 meters 210.
That sounds good, I think.
So Lynette, right now auto X, Y, and Z are on.
I know where this is going.
The altitude's dangerously low because Herc's looking downhill, so the DVL is real close. And you might, here, this, I'll come up a bit. Right? So it should be stable. That. If you want to move the vehicle, turn off auto XY, move it around, put it back in. If you want to turn the vehicle's heading, turn auto heading off. If you want to change your altitude so you can go in and out. You happy with that? So my back teeth are floating. I'm going to hand it to you and I'm going to run away for a minute. And I'll be right back. What could possibly go wrong? You've got more time in this room than anyone else besides me and Ed, so you'll be fine.
Source cam, pilot.
This might be a good moment to check in with our online viewers. We have a group that's joining from the Mariana Marine National Monument, so we're excited to have them join. The Center for Living Independence group is joining and following along with our dive. So we are here at Toto Seamount. We are about at 2,900 meters deep, and this dive started for us last night at around 8 p.m. So we're a little over 13 hours into the dive. Jacob got 2 rocks. We're feeling good about that. We slurped up a lot of shrimp, and the squat lobster have made another appearance, so I'm happy with that.
Yes, it seems like the squat lobsters are only in the south part where we saw them at first, but then, you know.
Quite a few here.
And Bill, you're joining us. You want to talk a little bit about our objective, kind of where we started on this dive and where we've gone, what we're searching for?
Yeah. Not super successfully so far, but yeah, the last time this site was visited
I thought this was this time.
was in 2003, and at that time this was a Japanese expedition with a manned submersible. They found multiple sites with these sulfur-rich or sulfur chimneys emitting white smoke, so acidic sulfur-rich vent fluids up to a temperature of 170 degrees C. So we are trying to find that vent field. We're pretty sure we're in the right neighborhood because there's lots of that white smoke in the water. There's some of it coming in from the left. But it's really hard to hunt down the source of it because as you get closer and closer, the visibility gets worse and worse, and then pretty soon you can't see anything. And so we kind of have to come at it from the right direction and have some luck. We're trying to do a search pattern here to find it, but the animals that live around the vents are also kind of an indicator of how close we are to where and how much warm water is coming out of the seafloor. So animals like the shrimp and the squat lobsters and the tube worms, those are all chemosynthetic, so they're just living around these seafloor hot springs. So the more animals we see,
Can I just sort of back up?
You can do whatever you want.
Closer we are to the
Whatever
vents.
I want.
Whatever you want.
Yeah, so we've been kind of making these traverses up and down this slope, and so now we're, you know, each time we move, we have to
that one there is you
orchestrate this dance with the ship, and
reverse cam, and then you have your
sometimes we have to
rail
wait for the
cams.
ship to move where we want to move. So that's kind of what's happening right now. But boy, our visibility is going down.
Could have been worse.
So
Mackenzie,
it's this mixed.
can you turn off the port and starboard lights?
Mixed luck, because it's good. Maybe we're getting closer to the source, but then we also
the less we
ruins
see
our visibility. So
you should be able to see a little better.
Just a hard place to work in.
Right.
It's not blowing it out so much with that light on.
Yeah,
Oh,
we're
okay.
in this sulfur-rich water, so
or
that's
if I
the
turn down
milky
slope, will
appearance
I
you see
really
on the
not be able
camera.
to see anything?
Right.
Sorry, I gotta turn this field out.
And the white clouds are actually,
say that again?
there's
Can I
tiny
turn, or
particles
if I turn
of
down
sulfur
slope, will I
in
really
those
not
white
see anything?
clouds. So that's
depends
what
on the
makes
slope.
cloudiness.
I tend to crab or whatever. You see your tether wraps there at minus 0.8, so you want to turn to minimize those. I'm not sure which way that is. I can never remember.
Okay.
You're fine now, but if it goes over 1, you'll want to turn the other way.
Okay.
The amount of sulfur on the rocks is another indicator. Like right here, there's not a lot of sulfur on the rocks. The rocks are, you know, dark looking. In other areas,
Looks like
all the
Anna's
rocks
headed
look
south,
white
so yeah,
or
that's the
pale
direction you
yellow
want to
because
go.
they're just coated in a layer of sulfur that's part of, you know, deposited from these hydrothermal fluids seeping out everywhere.
As you
So
probably know, when we're going downhill, we kind of
if
do the opposite
we're
of
not
uphill.
seeing that,
So
we're probably on
stay
the,
under, or
you know,
in this
outside
case,
the
a little
main
to the
area of
north of
venting.
Atalanta. So you're perfect.
So here we got some cloudy water, but we don't see a lot of sulfur on the seafloor. So
Oh gosh. Oh gosh.
There's a cliff. Jump off of it.
Should I turn off auto depth?
Yeah, I typically, if I'm hot dogging around, I turn all those off.
Oh wait.
Yeah, turn them all off. Fly that ROV, Lynette.
I don't know what I'm doing.
It's so the reason for that, it's much more responsive when they're off. So right now you have about 25% down trim on, so
Okay.
it shouldn't float up too rapidly.
Okay.
And when you come down, if you're facing downhill, I'd literally come down till I see a bump. You'll see the camera shake, then you know you're too low because you're touching. So if you're looking downhill and you're seeing 2 meters on your altimeter, you got that plus the slope.
Right.
And so it's hard to see.
Oh,
Yeah.
there it is.
Yep.
that's called a bottom integrity check.
Yeah, that's tactile feedback. And we use everything we can get here to figure out what's happening.
Goodness gracious. Okay.
Did you ever get a sense for which way the smoke is coming?
Oh, I don't know.
To me, it's like fog. So it's like, which way is the fog coming from?
Would it be better to face the slope and back down the slope?
Yeah, it can do.
At least we'd be seeing the bottom a little better.
Okay,
So you
turn this
the
way.
other way. Yeah, that's one turn there.
Go the other way.
It confuses my reptile brain because if you turn Atalanta, one thing happens. If you turn Herc, the other. I can never remember. You would think I would get it after all these years, but
It's complicated.
well, it's kind of like east and west. You know how often I've said,
So
you've had to correct me for that.
I get it wrong too.
I might be dyslexic.
So I think your heading should be northwest, and then we're backing down to the southeast.
Roger. Lynette's doing some tether management.
Oh, okay.
Whoa, I'm way up there. Goodness gracious. Well, I guess I have a bunch of slope behind me.
Yep, it can come down now, I'll tell you. Gently come down. I would say it's maybe 2 meters difference. Well, actually, if I think the slope's constant and I'm holding a heading, I'll come down and lightly touch your bump or get real close, and then I'll look at my altimeter, and then I'll know that'll be like zeroing.
Okay, sure.
Mental zero. I'm going to put the upper camera to mid-zoom, otherwise I'll get a text from Jonathan. You okay with that?
Yeah.
I'll put that one. So he likes it there for when we're, I don't know why. Hi, Dave.
Do we want to keep going this direction? Do we want to move Atalanta again?
I don't know. You're driving.
We want to keep moving
yeah,
southwest?
let's keep going down slope, southeast.
Southeast, sorry.
Yeah.
Yep. You're not the only one with an east-west.
Okay, southeast around 20 or 30 meters?
Yes.
Okay.
Please.
Dan, I've got a second dot on the Zeus lens now, actually a third, just for your awareness.
20225. In nav speak.
So Kyle, were you muted?
What's that?
Yeah, I think I was. I
Oh,
just
there
have
you
a message for you if you want to come check out from science. We have lots of
Oh,
scientists ashore joining us as well, sending in feedback from previous dives as
Okay.
well. So
Ah, all right. I'll hop over there.
Anybody know what intercom station Dan's on?
Yeah, well, I think it's nothing we don't know.
Oh, sorry, Anna. I was the one that was confused. They said southeast? Oh, yeah.
Hey,
So I
Dan,
get
are
those
you there?
backwards all the time. Yeah, I'm over here.
Southeast and southwest.
Just for information, I've got 3 drops on the Herc Zeus lens now.
Yeah, roger. Wish we had a windshield wiper cleaner. Looks like it could be hydraulics. It's an old machine. It leaks a little. How's our comps? Kenzie, have you checked those lately? We're still good? Roger. The main res is still up there in the 80s or 90s. The one on the.
All right, so just got some feedback from Dave Butterfield, who's a scientist ashore, telling us that he was reviewing the dive video from 2003 of the Shinkai submersible dives, and that the sulfur vents were relatively low flow rate vents, kind of, and in a bottom type, not too different from what we're seeing. So that's good to hear. Depth of 2937, yeah, that's what I remember. So we're a little bit deeper than that. On a steep slope. Yeah, so I think, you know, we're sort of in the right neighborhood, but it's been hard to pinpoint. I see some smoke down below us there. Maybe.
Okay.
Mackenzie's gonna come down on the winch a little for you, probably getting tight.
Okay.
We'll keep so since we're close or under Atalanta, we'll keep the delta up there around 20.
Sounds good.
And Mackenzie, if you look straight down with Atalanta's camera, you'll probably see Lynette under you. Right. That looks like an interesting cliff to go down.
Yeah, it's stressful. Do you want to take over? I would love for you to take over.
You're doing beautifully. Come on, you haven't lived till you've run both the vehicles into the same cliff.
Ask Dan to compute how many meters we are from waypoint 6. Stress him out.
Waypoint 6. I can't see waypoint 6. It's not on my sonar.
I know, that's why I picked it.
Oh, it might be up there.
Oh, I would say 10, 20, 30, 40, 50 meters. I've spent a little time in the nav chair. I know how to right click, believe it or not.
It's not easy.
No. I also failed 3 times in a row in math, so. Yeah, that's a nice geology there.
Would
Those smokers.
Would you guys be able to zoom in on that kind of spire feature to the top right of the screen?
Yeah. Let's fly up to it a minute.
The lasers are at the base of it now, that one.
You're, right now you're like 5 meters away from it.
On the right side of the screen is coated in sulfur here.
I use this one when I'm judging.
Okay.
Yeah.
And the way it's sticking out, you can fly right up and bump the porch against it if you want.
I don't want to.
Yeah, you'll be fine.
See how sturdy it is. Yeah. It almost looks like it's being, parts of it are either cemented together by kind of this coil of sulfur that's been like precipitating out, or I don't know, maybe some tephra as well.
Oh gosh. Oh gosh.
Deep breaths, Lynette.
Atalanta is right above Hercules.
Yeah.
So science team, what is it that causes that
sulfur
deposit to look this way, where it's almost like a fine sand almost?
Yeah, it's actually kind of a crust.
It'll look
it's
down
like,
some more. You might have to play with the slider.
Dairy Queen dipped cone. So there's lots of sulfur dioxide in the vent fluids that are seeping out of the seafloor here, and when the SO2 mixes with seawater, it precipitates sulfur particles, and if it's warm enough, then those are like molten little sulfur droplets that stick to the things around it. So that's how these rocks get coated in sulfurs. The SO2 is coming out of the seafloor. If it's warm enough, then it can be little droplets that then stick to the rocks. So a little bit earlier, we bumped a part of the seafloor where it was coated in the sulfur, and where the piece fell off, it was black underneath. So you can see that.
Oh, thanks.
The white yellow coating is just on the surface.
I think so, yeah.
Okay.
Well, all right. Speak out of turn.
All right, I'd say let's continue downslope a little bit more, and then maybe we'll jog a little bit more, like 20 meters in the southwest, and then go back upslope. I think maybe we should, after we do that, we should get up to about 35 meters, and then maybe contour around back to the northeast, since that seems to be the depth where those white smoker chimneys were found before. We should probably hunt around that depth contour.
It's a good thing Ann is here, because he lost me after southwest.
Okay.
Yeah, can I just confirm 20 meters southwest?
Well,
for this
let's continue downslope a little
You
bit
want to
more.
jump in?
Just keep going down slope in this
Yeah,
direction?
another,
Sure.
Okay, so southeast for a little bit?
yeah.
Okay,
And then after that, we're going to jog southwest.
okay, so
About,
20 meters
yeah,
southeast, and I'll check in with you
okay.
when we get there.
A science and ROV, I want to try and address an issue with our imaging system before we get trucking on. Picking up some, what appears to be fluid or gas bubbles on the primary lens.
Yeah, that's liquid CO2 droplets.
Okay, and they seem to be expanding as we ascend, as you might expect. If we tilt up, do you think they'll release, or that's just...
They're kind of sticky. They have a little bit of clathrate on the outside of it, like a ice
coating.
Yeah. So, and then, you know, as at the end of the dive when we ascend, it'll turn to gas and.
Right, just like chasing a bubble up.
Yeah. We've seen some of those floating around in the water at times.
Basically, Ed, they're slowly eating a hole in the $50,000 this lens. And at any moment, it's going to implode, take out the entire vehicle.
Dan.
And Bill, what did you identify those as? Just
liquid CO2 droplets.
Cool.
Yeah,
yes.
So there's CO2 coming out of the seafloor in addition to the SO2.
Right.
But at this depth, CO2 is a liquid instead of a gas. So they're little.
Oh man!
Occasionally we see them floating around in the water.
Well, we were able to get them off the lens at northwest of Fuku, some of them, by tilting up and sort of and maybe on the they actually sort of banged it a little bit, but it probably would be worth it to give it a shot.
Yeah.
Yeah, I've got 4 or 5 of them right now. The lower row has expanded when we ascended.
You could try.
Bill's probably right, but you got nothing to lose.
Yeah. So go ahead if you want to try it.
Yeah. Do you see what we're referring to?
I do, yeah. And I'm seeing we don't have windshield wipers on any of these.
No, because you don't expect to run into something like that.
I'm afraid of obviously the impact on the image quality, but I'm more afraid of having to remember how to compute partial pressure of gas at depth.
It's funny, my youngest last night was asking me a specific question about out here, whether the scientists used physics more or chemistry more. He's choosing a career path.
Depends on what you're interested in.
Yeah.
All right.
But it led to a discussion about pressure and temperature and chemistry, some of the stuff you just described.
I like your thinking with racking in and out, Dan, but those seem to be stuck.
I did rack in and out.
Yeah, that's what I mean. I like that idea, but
Oh,
just live with them.
Have moved.
Jonathan, watch out for your camera. I tried.
Is that as fast as it pans?
No, but it is a 20-year-old camera on a 20-year-old pen and tilt. And I'm surprised that it's still functioning.
The camera is on a sled that moves forward and backwards, and Dan did move it to the front, limited that to try to bump these off, but unsuccessful.
I can move it a lot more rapidly, but it always really scares me.
You have a little bit coming into Atalanta's danger zone up there.
Roger.
All right.
That is a red wall of death encroaching Atalanta's 20-meter range ring.
Sure is.
We are breaking the rules, Bill.
I think we can stop going down slope and now jog to the southwest.
Southwest.
Like, let's say 20 meters, and then we'll
go
go back up slope.
Roger.
Wow, there's still some little tube worms down here.
Yeah.
So, and you notice the sulfur coating, so, you know, it's just seeping everywhere here.
Yeah, but it comes and goes.
Yep,
I mean,
no, you're right.
some areas don't have any sulfur, and then all of a sudden you're back in it.
Which way is Atalanta facing? Yeah, if you move to the south, that'll move us away, right? Right?
Yeah.
Yeah. Yep, that should be a safe move. Yeah, so what I'm doing, I'm looking at Atalanta's heading, and it's looking that way. Here's where Rennie always does this. So it's looking that way, but it sees that target in front of it. So you got to do some mental math to which way is a safe way to, so if you move Atalanta on this nav screen, it's going to move that way. So
yeah, maybe a little more south than west.
Yeah. It's
I'm always impressed by your 3D,
say like
the
210
spatial
maybe.
relationships that you guys have to do. It's
painful mental gymnastics.
Oh.
The other thing I can do to make it easy is I can
not correct.
Actually, I can turn
try
Hercules.
grabbing this. Oops, you might have to move it back up.
So if I turn Hercules to the southwest in it, then we don't have to do mental gymnastics. And
like 210 should be kind of down in here. Yeah.
I'd say it's safe.
Yeah, I think that's good.
Bonk. Contact. So we're heading in the general direction on waypoint 2. I don't see any green squiggly lines in that area yet.
What would the green squiggly lines be?
On the ROV nav PC?
On your screen. I'm looking at a different one.
Yeah,
a different color.
You can pull
palette.
that up.
That's all right.
Pull that up back there if you want.
I gotcha.
Or squint from back there. But sorry, I lost the plot. We're headed
Southwest.
Southwest.
Thank you.
Yeah.
That way.
Correct.
Roger.
That's southwest.
Right. Or lower left.
Yeah. Have to go from 2D to 3D.
For anyone that is checking in with us here on our first voyage of the 2025 season, this is 171. We are currently in the Mariana Arc volcanic exploration mission here at Toto Caldera. And we are about 75 miles southwest of Guam, where we launched yesterday, and surveying the hydrothermal vents of this seamount in search of all kinds of unique scientific information for us to share with you. So we're glad that you're joining us from all over the world through our telepresence. And please send us any questions you have for our science and ROV team and video team who are doing an amazing job getting footage here. And we are thrilled to be able to share it with you.
Look at that rock.
Yellowy.
So, Rena, we did see a few of these big snails.
Yeah.
There's one there in the upper left, is it? Did you want to?
Nope, don't need them.
Don't
No,
need those
no
guys?
idiots,
Okay.
and they're always very peripheral.
Yeah. We've only seen a few, huh?
Yeah.
Okay. Yeah. So the snails you were talking about are smaller?
Yeah.
Okay. I haven't seen any of them, but
I
don't know
did
what
see
they
some
look
at
like.
the. I did see some at the beginning.
Huh.
Yes, and that's also interesting because that snail, geez, that one occurs on the back arc.
Oh.
Yes, and I'm just realizing we've never seen it on the arc before, the big one.
Oh, the white one.
Yeah, and but it's always way on the periphery,
Yeah,
and there's
yeah.
some argument as to whether or not it might be more deep sea than vent.
Right,
But
right.
they were really abundant. Do you remember the snail graveyard at Half a Day? Where the hairy snails had tumbled down a slope, and there was just all the dead snails, and it was away from venting.
Right.
They were all over that.
Oh, yeah.
As scavengers.
Right.
Yeah.
Huh.
And
there's our little squat lobsters swimming.
Coming out again. What is this this form we're seeing currently? I'm just looking through the fisheye camera too and seeing it's kind of like a ridge. What's the kind of structure of the part of the caldera we're currently surveying?
It's a little hard to tell without high resolution bathymetry. Somehow so more or less we're in the floor of the caldera. It's kind of oval shaped. Around the edge of the floor, especially in the northeast where we are, there's kind of an arcuate ridge on the floor that's some, I don't know how exactly it formed, some combination of subsidence of the floor or maybe some uplift and subsidence together.
Yeah.
Anyway,
up to Bill, I think. I lost the plot.
There's a lot of cracking along this ridge, and that's where all this hydrothermal venting is happening, so that somehow this structure is giving pathways for
stop for a minute.
the warm
I'm going to check my gauges.
hydrothermal fluids to come back up to the surface. So
Mackenzie, I'm going to turn off the downlight so you should get a good...
Hey, Science.
Okay.
Our ship is stopped right now. Do you know what direction you want to go from here?
more or less upslope.
Upslope.
Yeah.
The res.
Okay, did you want to take a step sort of west or east and head upslope?
Yeah, we were trying to step southwest and then go upslope. So we've come a little bit southwest. We could probably come a little bit more, down to where the cursor is on the high pack.
Okay.
About another 10 meters or so?
Yeah, and then start heading upslope, which is northwest.
I'm sorry, can you say again? A step west, 10 meters?
Yep.
Okay. You okay with that, ROV?
Yep.
Okay.
So along this ridge, there's lots of steep slopes, lots of,
looks like
wow, it's kind of a big pillow.
Yeah.
Looks like
maybe
all by itself.
All right.
The little kid in me
Whoa.
You can fit that on Herc, right?
just wants to roll it down the hill.
Yeah, so it's
Cool.
um,
I'm gonna reset
this
DVL.
ridge
Unless you want to do it.
seems
Do
to
you
have
wanna do
some
it?
big fissures running through it, so that's some of the
So we
steep
just want to make sure
slopes.
that auto XY is not
And,
on, and it's not, so we're okay.
But It's a little hard to piece together just from the video.
Cool.
Some kind of mat there on the bottom, wafting in the thruster wash. Sorry, I stirred it up there. And I stirred it again. Yeah, it's because there's a clip to my left.
All right. Yeah, I think we can start heading up slope from where you are.
We have another 4 meters on the vessel move.
Okay.
And then, yeah, I mean, upslopes kind of back the way we came. You want to zag or zig?
Or, you know, drive northwest, more or less.
Northwest, right.
Yeah. That should also
I'm
be
going to
upslope.
adjust Atalanta's heading a little bit.
All right. I'm going to come to the northwest of you, so.
All right, so this time we're gonna, I think we'll come up to a depth of about 2935, and then we'll want to kind of contour at that depth back to the northeast when we get up there.
Working on it. There was this big rock in my way. And there's something to my left here I keep bonking into.
That's, yeah, northwest should be the upslope direction, so.
Yeah. This way.
Yep. Skydivers. They've got to be saying, wee! Oh, is that a clam shell?
Looks like it.
Yeah, it looks like it.
2 of them.
Oh, yeah, Verena.
Oh, well, we'll take a still shot.
Can we, yeah, let's have a look at those. If they're alive, I think we would like to sample those with the one of the scoops.
Yeah. I can zoom in on Ed while I'm looking for a little pink snout somewhere.
All right, coming in, yeah.
Nice. That looks alive, doesn't it? Yeah,
I do.
All right, let's scoop those puppies.
I could scoop them or just pick them up. You want them undamaged?
I think probably.
You want the shell?
I'm just going to reset this again.
Vereen is the one to answer that, so we might have to wait till she returns. But I think we had talked about this scoop, so I think that's probably what we want to do.
Roger.
Do you want to put them in the box with the chimney or the rocks?
In the interest of time, I can.
With the chimney's fine, I think.
Pile them up.
In the front box.
Okay.
Get a shot of all of them in situ.
I'm going to porch out, Ed. So.
And we're back.
And
oh,
then
wow.
I'm going to box open.
What about this group on the left with the snail?
And then we twacked the camera.
We twacked?
We twacked him.
It's twoo.
Which of your 3 toys do you want, Bill?
Let's go with the one on the port side, the big scoop.
Right.
Or do you think, or is the shovel better, or what do you think? Do you have a preference?
Yeah, I'll go for the shovel.
Yeah, the scoop versus rock sometimes doesn't end well. Is that snail of interest? Oh, we need we need a marina. I assume that's a snail.
Okay.
You want to do a tight zoom there, Ed, on the volunteer? Tilt down a little for you.
Man, let me see if I can clear that up a
Twofer?
little. Oh, it goes right there.
Oh, get greedy, going for 2. And
Pan the camera a bit.
sure. 2's not happening.
Polychaete?
Oh, it's a polychaete, yeah.
You want a close shot of that, Dan?
Happy?
Yep.
That one in the box? You want the rocks too, or?
There's
not much I can do about it.
Yeah, whatever.
Yeah.
Whatever you need to do to get the clam in the box.
Yeah. Okay, go ahead, Ed. I'll tilt down for me on the camera and show me the box.
We'll probably want to get some more. Oh, I thought you were going to empty the scoop in there.
I should go away more often.
Do you want more than one, don't you? Yeah, I'd say 5.
Really?
Yeah. Sorry, I thought you were going to just empty the scoop.
Oh.
Should have caught you.
Spaz jaws. Sorry. That's
we could also do one of the other scoops if you want to switch, but
I can empty it.
yeah. So I'd say we're aiming for 5 clams.
Roger, 5.
Oh yeah, you can dump the rest of the rocks back on the seafloor. Yeah, perfect.
I'm just gonna move the vehicle here. So.
Yeah, about that big, I'd say.
So are these clams the ones that are unique to the back arc that you find here at Toto?
Okay, Ed, you can zoom in there, please. Zoomy zoom.
Yes, I can.
Beautiful, thanks.
Oh, yeah.
No, in terms of the Mariana system,
Ooh, look, there's snails too.
these clams are unique to the site.
I didn't see the snails.
Yep. They, a couple were collected on the Japanese expedition, and they were identified as a clam that is known from cold seeps.
Oh, really?
Up near, let's see.
But nowhere else in the Mariana system.
Nowhere else in the Mariana system.
Huh.
I think they're known over by Okinawa, but I have to go and check.
Uh-huh.
They were first found by a Japanese-French expedition back in the mid-80s.
Huh.
In this, but I'm just blanking on where. So it's
Okay,
odd.
I
This is such an odd site biologically, because it's a mix of a mix of biota from different areas. Actually, well done. That's,
okay, so we're getting one more after this. Don't let go of that scoop.
Roger. Watch out for that box. Yeah, still getting used to using the wrist on this thing in position control. Our old arm,
okay.
we could only use it in rate. Okay, do you want to zoom in? You want a bonus snail?
No. No, we're unless the museum really wants it, but
Sean wouldn't mind a snail.
Sean would like a snail. Okay. They're quite lovely. I agree.
Bonus rock as well.
Sure.
Yeah. Nice.
Do you want all the way up?
Okay. Yeah, now you could put the scoop in if you wanted to.
Roger. Bombs away.
So Verena, what is it about the clams that allows them to be so unique in this one environment? I mean, where do they come from, and why only here?
I have no idea.
What's that, Mackenzie?
It's a short answer. I don't understand why this site seems to have accumulated species that we're not seeing elsewhere on the arc or back arc.
All the way closed.
And yet there's species that are found over near Okinawa, and a few species also found near Tonga. And so it's almost as if it's a crossover region. But this clam is from the Nankai Trough. So it is Northwest Pacific. Why it's landed here is a good question. There's a lot of species in this group of vesicomyid clams that are found all around this Northwest Pacific region. Many different species. So it was, but none
Does it have to do with that?
other ones on the Mariana Arc. So what this clam, it's fairly large, as you can see. It's about 10 centimeters long. The flesh is red, hemoglobin again. It has no gut, so it is what's called a holobiont, which means it's an animal which has a complete symbiosis with with microbes, and together they form a unique sort of biological system, which is referred to as a holobiont, which is an interesting term. But so the symbionts are in the gills of the clam, and the gills take up oxygen, carbon dioxide, and the hydrogen sulfide from the vent water. It's shunted into the bacteria, but the bacteria have special transporters to take those compounds across their membranes, and then the sulfide and the oxygen combine. They are the mediated oxidation happens inside the microbial cell, where ATP is produced from that reaction in a cascade of biological reactions that is very similar to photosynthesis. So instead of light photons coming in, what we've got is energy from the reaction of
whenever
of
you
the
guys
oxygen
are ready.
with the sulfide.
I think we're
So
going that
that's
way,
the
right?
part that's different. But then the cycle to turn that into energy molecules that then take carbon dioxide and convert it into glucose, which is sugar, which is the basis of building food. That is very similar to the photosynthetic system. And so these that glucose now, or the food molecules then leak out of the microbial cells into the host cell, and then the host is basically feeding off those products. So this clam wants to be somewhere where you can get all three of those compounds, oxygen, sulfide, and carbon dioxide. The foot takes up the sulfide, the gills take up the CO2 and the oxygen, and then the rest of the body is working to get it to the bacteria and then the food out again. It's an incredible system, and it's
fascinating.
So I'm gonna try something here. That little patch right there with the rock and the stuff growing on it and the gold. I'm gonna see how close I can get your stills camera to it for a close-up photo. Like that's as close as I can get. Oh.
got little worm tubes coming out of the sediment here.
Yeah, sorry. Not a stable landing platform. It's interesting, I just touched it and it
Yeah, so here's another area without the sulfur coating over everything, just loose rocks and rubble coming down slope, but we've still got the squat lobsters scattered around, so we're not too far from the venting.
Is the sulfur from the venting or from an eruption?
From the venting, yeah. The hydrothermal fluids seeping up.
Comes out as a liquid and turns into a solid?
Yeah. Well, it's
more or less.
The SO2 is dissolved in the vent fluid, and then when it mixes with seawater, it creates tiny sulfur particles that then, if the vent fluid's warm enough, it's little droplets that stick to things like rocks.
Noticing a lot of anemones here as well.
Yeah, yeah.
Are they also feeding off of that sulfur in the in the water, or what's their food source?
Their food source is either little tiny plankton or even just detrital organic material, but they're sort of capturing whatever they can that's has some sort of carbon and nitrogen in it that they can use. I don't know what these are. We've not studied them, so it's a bit of a mystery.
There's also a polychaete in the water to the right of the lasers. I guess he just landed over there.
Which one are we going on?
Yeah, to the right, one of those white guys.
Like way far to the right?
Yeah. Between the lasers and the squat lobster. Yeah, see him moving?
Oh, yeah.
Those are two polychaetes.
Oh my goodness.
Or are they?
Are they?
One of them is, maybe. I don't know what they... No, I'm not sure.
No.
I thought one was swimming, so...
Must investigate. You can push in there, Aaron.
All right.
Push all the way into the lasers at the top of the screen.
I guess not. It's just a flocky particle.
Yeah.
Huh.
Well, okay, where's that come from?
Yeah.
So I was sitting there for a minute trying to work out which way the wind's blowing.
Mm-hmm.
And go wide again, Aaron.
Yep.
Sorry.
Oh, there's the polynoid.
Oh, okay.
It seems like it's coming up this structure here.
There was a question in the chat about the difference between squat lobsters and yeti crabs.
Oh.
I'm not familiar with yeti crabs, but if you have some insight on that.
Okay, actually yeti crabs are...
Full zoom there, Aaron.
Yep.
Sorry, I'm just... I'm just blanking. They are...
Are they not?
Just a moment. Yes. Hearing.
That guy's cute.
Okay. So they are related in generally in the same larger group, but yeti crabs are very specific to a region in the southeast Pacific, on East Pacific Rise, down pretty well south, and then down in the South Georgia Arc near Antarctica. There are similar kinds of crabs that, but they're all in the same group that is kind of between crabs and lobsters, I guess. They actually are more related to hermit crabs than anything else.
Did we just turn off the lights?
And the yeti crabs
I should have warned you.
farm bacteria on their claws. That's why they look like what they do. What we're focusing on right now is a very lovely polynoid polychaete called a scale worm. And this looks like it is a vent one because it's got hemoglobin in it. So that's why it's pink. Oh my goodness, I'd love to have him.
Really?
Yes, I really, really would.
Really? We can slurp him up.
He's an amazing PhD student who's just finishing up her thesis at Scripps, and she has worked on hundreds and hundreds of polynoids to try and sort out the systematics, the phylogenetic relationships of many of these that are found at hydrothermal vents.
Actually, Aaron?
Yep.
Because it's been
you put,
very difficult to do it morphologically.
Never mind.
So Avery Hawley with Greg Rouse has just published a paper redescribing and renaming all of these animals. I look at this and I'm going to guess it was what we used to call Branchinotogluma, possibly Marianica. But it's now been placed in a new genus, which I'm struggling to remember, which might be Themis.
And is that all based on genetics, or?
But it's based in genetics. But what was really interesting, Bill, was that